So, hello everybody, welcome to the cluster seminar. And this week it's Mental Health Awareness Week at the University of Constance, and this coincides with a mini talk series on stress, the effects of stress, and also the protection against stress, and who would be better to enlighten us on this important topic than our cluster PIs who have been working on stress through the collective lens. Today we will have Jens Pruessner giving the first of these talks, and then Petra Wirtz will present later in the semester on a related topic. So without much further ado, Jens, take it away. Thank you.
Thank you, Alexandra, and thank you for being here. I'm happy to have the opportunity to talk a little bit about the topic of stress and try to incorporate some of the findings that we've contributed to this field over the years. So a little bit of a mix of general information about what stress is, what it does, and some of the research findings we've been able to contribute. The overview I've set out is also very straightforward. First, we'll talk a little bit about generally what stress is, how we can measure it, what are markers of it, and then talk specifically about what it does in the brain and what it does to your body. The term originates from physics, and it basically was meant to mean how much pressure you can put on an object before it breaks. And this was a nice to visualize concept which was transferred to biological systems. How much pressure can you put on an individual before it breaks? Hans Selye was the one to start this. He's really the grandfather of stress, and his 1936 book, the cover shown here, The Stress of Life, really made him famous. He was a researcher from the University of Montreal, studied stress all his life. At the very end of his career, he then published another book which was called The Stress of My Life, to illustrate how much he also had to endure as a very sought-after researcher. So the confusion of cause and consequence, which typically is not differentiated when you talk about stress, is really that the stressor is the cause of what stresses you, and the consequence is the stress reaction that you experience, changes that happen within your body that basically then constitute the stress response. From that definition, we can start to ask what constitutes a stressor. So what is it that then actually can stress you? And this definition has really evolved over the decades. And coming back to Hans Selye, which we just heard of, from his 1936 book, he defines stress as a strong demand on the body, very general, not very differentiated, which was then refined and revised by Mason in 1968 in a big review, where he basically identified these characteristics of ego involvement, novelty, uncontrollability, unpredictability, and anticipation of negative consequences, which was really a huge step forward. It helped within the human stress field to much better identify what actually might be stressful and what might not be stressful. But I think the optimal description was then really added by Dickerson and Kemeny in a review in 2004 in Psychological Bulletin, where they coined the term of social evaluative threat and uncontrollability to be the prime characters of causing a stress response or identifying a stressor. And the Psychological Bulletin review was really nice because they compared effect sizes over the years across all the studies and could basically go from passive tasks to motivated performance, adding uncontrollability, and then basically have motivated performance with the element of social evaluative threat and uncontrollability causing the strongest reactions. Now, of course, you might ask yourself, what are situations where you have a motivated performance with social evaluative threat and uncontrollability? They coined the term a social self-preservation system to illustrate what they meant by social evaluative threat. And the idea is that we humans are basically unable to live in isolation. So we rely on our peers, we rely on other people to strive. And this has the consequence that we are also having a certain position, a social position within the hierarchy. And the social evaluative threat is basically any situation that now threatens our position in our social hierarchy and makes us lose our status. And Dickerson and Kemeny consider this to be a really important aspect of stress, because if you lose your status, eventually you might be rejected from the group. And evolutionary, that meant you were threatened to die because you rely on your peers, you rely on your social environment in order to make it through life. So they argued that this social status is really something that is really important to our survival. And so in the laboratory, what has been contributed by my doctoral supervisor, Clemens Kirschbaum, in 1993 was this Trier social stress test as one translation of a situation that contains social evaluative threat. And it's basically a free speech in front of an audience about a topic that is unknown to you, combined with mental arithmetic after 5 minutes, where you have to count down from a 4-digit number in steps of 13 until you reach 0. Whenever you make a mistake, you are asked to start over. And this is, of course, something that can be thought of as embarrassing. And especially if there's a larger audience, then you can imagine how performing poorly might then induce this kind of threat to your ego, to yourself, in terms of the social evaluation that you're experiencing. In lectures that I give as part of seminars, there's more time, so I always sometimes show clips from YouTube where you have moments in history where people have really, like embarrassed themselves in front of a huge group. One of the better examples is Rick Perry, when he made a governmental a bid for a presidential candidate in 2012 in front of the nation. So 7 candidates were basically presenting their program, and he had long made the claim that when he became president, he would do away with 3 departments that he thought were wasteful and to save the American taxpayer a lot of money. And in front of the whole nation, he was then unable to recall the 3 departments that he wanted to do away with. And the interviewer basically pressed him on, so you can't really remember the 3 departments you want to do away with? Very embarrassing. The whole thing only lasted 50 seconds, but basically effectively removed him from the list of candidates. Saturday Night Live, which is the comedy show in American television, made a 5-minute clip out of that to basically illustrate how embarrassing that was. I encourage you to watch it. It's really very funny. Of course, it wasn't so funny for poor Rick Perry. When you experience a situation that constitutes these elements, you basically get a stress reaction that now activates your energy systems. And I would think of stress systems mostly as energy systems, because what really is happening is that your body increases the availability of energy in response to the perceived increase in demand. If something is threatening, your system prepares for coping with that by making more energy available. And in overview, this is a picture from a review by Ulrich Lai and Herman in Nature Reviews Neuroscience, which I really like. You have the HPA axis as the major endocrine axis, and the autonomic nervous system with its sympathetic and parasympathetic branch that respond to this threat by basically changing their activity. And there's a certain timeline where the parasympathetic nervous system is the fastest to react to a stressor. It basically reacts instantaneously within seconds. It's the vagal withdrawal that leads to an immediate increase in heart rate. Then there's a sympathetic nervous system that kicks in with adrenaline and noradrenaline. That is second in line, takes from seconds to minutes, leads to change in blood pressure and also heart rate. And the HPA axis, hypothalamic-pituitary adrenal axis, is the last to respond, but that actually takes many minutes. Its last to respond was the final product, a hormone, cortisol, in humans, that has multiple effects in the body, which are mostly associated with all the negative effects that stress is also said to have. If you look at cortisol, this is the typical timeline over a 24-hour period. So it has a strong circadian and ultradian rhythm. So you do see that there's an overall circadian rhythm, with the peak around awakening and the nadir around going to bed. And this, of course, makes sense if you think of cortisol as an energy hormone. If you go to bed and want to sleep, then you shouldn't have lots of energy around. And in the morning when you wake up, well, then, of course, you need to be able to attack the day. And this is where you basically get the most energy from it. You see this little blip here that is superimposed on the circadian rhythm. And this is basically the response to awakening. And this was work that I contributed as my PhD work, where we basically could show that this cortisol awakening response seems to be very robust, happening across life, where you do get this additional doubling of cortisol levels within the first half hour, roughly 45 minutes in women, to the awakening response. Stress reactivity and cortisol regulation is so important because it has a strong link to health and disease effects. A lot of the modern illnesses that we know of physically, hypertension, coronary heart disease, and depression are linked to a dysregulation of cortisol. Within the psychological domain, there's also a number of illnesses like burnout, chronic fatigue syndrome, and fibromyalgia, which are believed to be related to a dysregulation of cortisol. How this exactly works is something that I'm now going to detail a little bit on for both the brain and the body. But let's take a look at first some of the effects that acute stress can have on the change in cortisol, to see what profound effects a stressor can have on the activity of this axis. So you see here that with the TSST, so this triosocial stress test, speaking in front of the audience, you get this huge change in cortisol levels within 20 to 30 minutes to maximize the stress response. And the triosocial stress test also extrapolates to heart rate changes, so it's including the autonomic nervous system, showing that when you are acutely stressed, you get both the autonomic nervous system and the HPA axis to respond. This, I think, is nice to illustrate how you have this timeline present in the responsivity. So we basically see the onset of the stressor here, and over the first 2 minutes, illustrated here as the heart period, you see this immediate increase in heart rate, so the drop in heart period. That's the vagal withdrawal, where basically within seconds you get a very strong responsivity from that. Then alpha amylase, as an somewhat noisy or dirty marker of the autonomic nervous system, you see reacts within minutes to then increase in response to the exposure to this social stressor. And cortisol, with the open bars here, takes longest to respond. So really, we're talking here 20 to 30 minutes until you see a peak change in this effect. We've shown before that these systems are interrelated, so that the magnitude in the response of one system with a lag is linked to the magnitude of the response of the other systems. So they basically talk to each other and synchronize their responsivity given the different timelines. And the link between amylase and cortisol is around 20 minutes at its peak. So cortisol is basically 20 minutes lagging behind the maximum release of alpha amylase. Now, we do have, again, from the review by Ulrich, Lai, and Herman, different ways to activate these two energy stress systems. We can have bottom-up stress signals that come from the body, signaling an homeostatic imbalance, pain or inflammation. They go through the brainstem to then basically coordinate and negotiate the activation of these axes. You also have the top-down, and those are stress signals that are basically processed in the prefrontal cortex, where you have to evaluate what's coming in and decide that this is something that you experience as stressful. And this evaluation and decision is, of course, something that now also implicates the rule for interpretation and for inter-individual variation. So not everybody responds the same way, depending on your previous experiences, your beliefs, and so on. So this is a really important point, because it basically leaves room for variation across individuals where the same event, the same stressor, does not have the same effects. So this basically goes into the perception of the situation as threatening, and this is something that you need to evaluate. And this is where the theory of primary and secondary appraisal comes in by Lazarus and Folkman, who basically say that something is happening in an environmental event, and you need to first look at your beliefs and commitments to decide whether you find this situation to be benign or important. And if it's important, it might pose a threat. And then there's a secondary appraisal where you basically compare the demands of the situation to your available resources. And if the demands are higher than your available resources, this is threatening. If the demands are lower than the resources, this might be a lot of work. You might be challenged by it, but you feel like you have what it takes. And then you can basically deal well with this situation. And this, I think, is also something that, especially if you look at stress from the context of Mental Health Awareness Week and what can you do, where many of the stress resilience programs come in to say that this is something where you can directly work at. How you evaluate the event and how you bring in the assessment of your own resources is changeable. If you believe in yourself that you have what it takes, you are likely less stressed by the same event than if you feel that, oh my God, I'm going to die. And there are many examples for this. One that I particularly like is the river rafting, where you have two coaches guiding you through these dangerous waters. The first one is really experienced, has a lot of years doing this under his belt. And whenever you cross a turn and there's an dangerous looking rock ahead of you, he basically reacts by saying that everybody stay calm, okay, let's see, you do this, you do that, and you feel like in very competent hands. If you have the unexperienced guide who is coming around the turn and sees a dangerous looking rock ahead of you and says, oh shit, you might feel less secure about the whole situation and might feel that this is more of a threat, and you are more likely to experience a stress response. We contributed to this question of inter-individual variance in the presence of the identical stressor over the years. And I'm just going to show you some examples of factors that seem to play a role here. Very early on, we looked at the effect of self-esteem, and self-esteem is, I think, very approachable as a concept, because you can think that if you are high in self-esteem, you likely might look at the available resources also to be higher. So here we basically had 4 groups. We pre-screened them for their self-esteem levels and basically split them into 2 groups with high self-esteem and low self-esteem. And also we exposed them to a computerized mental arithmetic task where the tasks were either rather easy or so difficult that you were doomed to not perform very well on them. And you can basically see that over time, the only group that really responded with an increase in cortisol was the group that had the low self-esteem in the failure condition. So the combination of a really challenging task together with the low self-esteem basically made them believe that they didn't have the resources, the necessary things to cope with the situation, and then responded with the activation of the HPA axis. The hard task alone in the presence of self-esteem high did not do that. And the high self-esteem in the presence of failure also didn't do it because they probably felt that they were still under control and could cope well with this situation. Similarly, when exposing subjects repeatedly to the same social stressor, speech in front of an audience and mental arithmetic 5 days in a row, we could show that only those subjects that had low self-esteem, those were the ones that we deemed or coined high responders, basically showed a much stronger response on the first day, which then also didn't habituate in the subsequent days. So upon repeated exposure, they continued to respond very highly to the same stressor, whereas the other ones that had the high self-esteem did show a significant response on the first day, but this habituated on the second day, so they didn't feel that this was threatening to them anymore. Sex difference is another interesting topic, and this has less to do with a personality characteristic, but the sex difference, so the biological difference between men and women, really seems to interact with hormonal differences between men and women. And this was contributed by Brigitte Kodielka from the University of Regensburg in a very nice review paper, where she could basically show that men have the highest responses to this social stressor. And then depending on the amount of estrogens circulating in the women, so whether they are in the luteal phase where estrogen is low, in the follicular phase where estrogen is high, or if they use oral contraceptives containing estrogen, where estrogen is basically highest, you do get lower and lower cortisol responses. And this has to do with the fact that the HPG axis interacts with the HPA axis. So the activity of the HPG axis basically inhibits to some extent the activity of the HPA, so that women, when they have maximum estrogen, have the lowest cortisol stress response. And again, biologically, difference between the biological sex. There are also, one of my favorite studies on the stress response are gender effects, differences between men and women that probably have more to do with sociocultural effects. And this was a study contributed by Thomas Klauer and Clemens Kirschbaum, where they basically tested men and women. And they had 3 groups where they basically had no support by a stranger, or support by their romantic partner. And for the men, if they had no support, they showed the normally strong, robust stress response. If there was stranger support, they had a somewhat reduced stress response. And if there was their romantic partner present, that really worked very well. So they had a much diminished stress response. However, in the women, they had the originally lower response, as we just saw, which didn't really change much with the strangers. But if their romantic partner was present, it actually made it worse for them. So they didn't have a benefit from this situation. And of course, you could muse why that is, that men benefit from the presence of their female partner, but females seem to do the opposite in the presence of their male partner when exposed to such a stressful event. Okay, let's start looking at some of the effects in the brain. And this is relevant because cortisol is a steroid hormone, and only steroid hormones can actually cross the blood-brain barrier and then be active in the brain. So there are a lot of glucocorticoid receptors in the brain where cortisol can have effects. And a former postdoc of mine, who is now a professor at the University of Hamburg, has, together with Oliver Wolf, worked for much of his research career on the effects of stress and memory. Lars Schwabe. So in general, you can show that if you expose people to this typical standardized social stressor, you basically get an impairment in stress retrieval. So in retrieval of information that was present at the time of the stress that is maximally 90 minutes after the onset of the stressor. So the conditions that have the stress neutral and stress negative information is most reduced 90 minutes post. And in a review that he contributed in 2012, he basically put this together in this graph, which I also find is very informative, where you basically see that the memory formation mode, the retrieval of information is suppressed, while the encoding of information just before the onset of the stressor seems to be facilitated. And in the memory storage mode, there's also a suppression. So the reconsolidation of this information that is typically happening is then also suppressed. So you are, if you want to put this together, likely changed in the activity of your brain such that you will remember well what stressed you. So this encoding of the stress relevant information is enhanced. But anything that you had learned before that you now want to recall, that is impaired. So you have a hard time of recalling already previously learned material. So for anybody having still to take any exams, the idea would be don't be stressed at the time of taking the exam, because you will have a harder time of retrieving the previously learned material. You will, however, experience and form lots of memories about the exam itself. So you will remember the examen very well. And we could show that this reconsolidation impairment that I just mentioned, you can also induce using a pharmacological agent, propranolol, and we could show that it involves the limbic formation, especially the hippocampus, and the hippocampus is, of course, our memory manager, if you like. So lots of memory formation goes through this particular structure. And that was the time when we were really interested also in studying the acute effects of stress in the brain. So we thought of something to actually induce stress inside the scanner, which is not trivial because you now know that there's a social evaluation taking place. So in front of an audience, ideally, is what causes stress. If you are locked away inside the tube, inside a room, there isn't really much space for an audience. So we had to think of something that would allow us to induce stress inside the fMRI scanner. And people before us had done pharmacological manipulation, as did we, but that doesn't really reflect the brain activity associated with HPA axis activation. So if you want to know what actually triggers the activity of the HPA axis, it's not enough to simply pour glucocorticoids into the brain and see what's happening. You actually want to know what caused the HPA axis to activate. So we had the challenge to come up with something that was socially evaluative inside the scanning environment. And the PhD student of the lab at the time, Katarina Dedovic, together with myself, then came up with this idea to create the Montreal imaging stress task, where based on the triosocial stressor, you have this challenging mental arithmetic, which is presented to you as these tasks, and you have a rotary dial to respond by using the 3 mouse button, left, right, and then confirm. And importantly, we brought in what we thought as a means of social evaluation. So this performance bar shows the average performance, and it shows your performance. And you're being told that you should try to be in line with the average performance so that we can use your results to compare with everybody else we've already tested. Then we let this run for 5 minutes, and this, of course, is manipulated. So the difficulty of the mental arithmetic adjusts in hardness and time to your performance. So you can try as much as you can. You will not get more than 50% correct answers. And we do this 5 minutes, you're in the red, we stop the scanner, we go in and we say, well, this is very surprising because everybody else seems to be able to do that. And the technician and the assistant, everybody there is really wondering, and this scan costs $500 an hour. So can you please try harder so that we don't waste this? And then we go out again, let them try as hard as they might. And of course, they still come out with the poor performance. And we do this one more time, and then we relieve them and say it was all a hoax. Sorry about that, but we needed to stress you. And this works, but it works only in about 50% of the subjects. Basically, the 50% that respond to this, that are competitive, that believe in the cover story, 50% do not. So we do get pretty much across studies very clearly this distribution of half of them shows an increase in cortisol and HPA axis activation, half of them does not. What is nice about this, however, is that you can now see what happened in those that responded in the brain activation to those that didn't buy it and didn't show a response. And what we could show there basically is that there's a very strong deactivation of limbic system components across the board. And I think best illustrated really in this PET study, even though these are red, normally associated with activations, those are negative. So basically it's the amount of deactivation, and deactivation means less active during the stress condition compared to the control condition. So really a shutting down of these areas. And you see here that it involves, originates from the orbitofrontal cortex, but then stretches into the medial temporal lobes, into the hippocampus. In the fMRI study, it's a very similar picture. So you see that it stretches into the hippocampus and deactivates this interior portion of the hippocampus. And the deactivation is linked to the amount of cortisol that is being released. And I think this does make sense from the perspective of what is the role of the hippocampus in relation to the regulation of the HPA axis. So the acute stress leads to an inhibition of the hippocampus, but the hippocampus in itself is inhibiting the HPA axis. It's like a brake on the HPA axis. So when stress now inhibits the hippocampus, you basically take the brake off, and this taking the brake off is something that then causes the activation of the HPA axis. However, since the HPA axis is activated by inhibiting the hippocampus, this then comes at the cost of memory processes. So the memory effects that we saw earlier might be related to the acute inhibition of hippocampal activation when there's an acute stressor. It could also explain why what caused the stress is better memorized, because until the onset of the stress event, the hippocampus was still active, and that can lead to less interference with subsequent events which are not processed, so it gets memorized better. Okay, let's switch to what are chronic effects in the brain. And this, I think, is an also very interesting area of research where for a long time it was dominated by the idea that cortisol might lead to neurodegeneration and lead to atrophy. And this comes from the observation that chronic cortisol exposure is shown to have neurotoxic effects, especially in rodent studies. And there are numerous studies which have also been well replicated where this could be shown. If you have chronic excessive cortisol exposure in rats, you do see damage and atrophy of the brain cells. So this increased risk for neurodegeneration that was observed in rodents was now the idea, or that was contributing to the idea that you might see neurodegeneration in humans because of high levels of chronic stress. So if you experience too much stress in your life, that basically puts you at the risk of atrophy and eventually dementia as a consequence. And this theory was coined the glucocorticoid cascade hypothesis by Robert Sapolsky. I never worked with the guy, but I still have to show a picture of him because I envy his hair. The stress that basically goes into the brain, crossing the blood-brain barrier, then would lead over time to an atrophy of brain cells. And Sapolsky believed that this was due to a modulation of mRNA, excessive excitatory amino acids that get potentiated by the effects of stress, and in inhibition of glucose transport. And in a science paper that he published in 1996, he could basically show that across conditions where you have very high cortisol levels, so PTSD, Cushing syndrome, and depression, you basically get an hippocampal volume effect, so reduction of hippocampal volume as a consequence of either excess cortisol levels or exposure time. And I think this was basically the high time of this hypothesis, where people really focused on, okay, is this something that we need to be on the lookout for? Do we somehow need to block the effects of glucocorticoids in the brain so that any high-stress manager is not doomed to get Alzheimer's in his 50s? So this is basically the detailing of what Sapolsky believed was happening. The modulation of mRNA, inhibition of glucose transport, and the potentiation of excitatory amino acids. However, and I think this was an so far still overlooked article by Swab, Bao, and colleagues in Aging Research Reviews in 2005, they could convincingly show that the hippocampal loss, so the reduction in hippocampal volume, is reversible even in Cushing syndrome. And Cushing syndrome is basically the highest cortisol levels you can get because of a tumor. The HPA axis is maximally active all the time. But if you stop the overexposure with surgery, the reduced hippocampal volume basically bounces back, suggesting that it's not really atrophy, but the volume change that you observe with structural MRI is really due to a change in water and electrolyte content. So they have shrunk during the time of glucocorticoid attack, but once that is over, the cells can recover in volume. And the other, I think, convincing argument is that those people that have long bouts of hypercortisolemia, so untreated Cushing for years or decades, have also no higher incidence of Alzheimer's disease, suggesting that it's not contributing to a reduction in volume. What I do think is true, however, is that you do see developmental effects present that have an effect on the formation of the central nervous system. And this goes in line with populations that are known to be more vulnerable to stress-related disease, show a dysregulation of the HPA axis combined with larger amygdala and smaller hippocampus, which for a long time, when all of this is correlational, of course, when these events came out, they served to support the glucocorticoid cascade hypothesis, because you had this stress dysregulation and you saw the smaller hippocampus, but you didn't look at it from the perspective, oh, maybe this actually happened early on in life, and once it was present, it caused these correlations without actually the cortisol being the cause for or the reason for these lower structures or smaller structures. So this is likely a consequence of CNS formation during critical development periods, and hence the concept of early life adversity programming your brain to have certain structures in certain sizes that has an effect on HPA axis regulation. And we contributed a number of studies over the years to basically show that this might be the case. Of course, all of this is correlational, because in humans you couldn't experimentally manipulate early life adversity, but you take validated questionnaires that look at critical events during the first 16 years of life and see how they associate with either cortisol regulation or hippocampal structure. So Veronika Engert, a PhD student at the time, who's now professor in Jena, showed that the level of maternal care as one proxy of early life adversity is related to the cortisol levels, even repeatedly. So there seems to be a systematic association between maternal care and the activity of the HPA axis in adulthood. Perhaps the I find personally most convincing contribution was in a cooperation with Annette Völlmin, PhD student at the time, at the University of Basel, together with Jens Garb and Klaus Bader, who could basically show that using the concept of ACEs, adverse childhood experiences, you get a dose-response effect, where the more of these adverse childhood experiences during critical development periods you have, the more pronounced your HPA axis is affected. So you do get basically more and more effects from more and more of these adverse experiences on the regulation of the HPA axis. And this also generalizes over to the autonomic nervous system, because these are correlated. You also see this in heart rate effects. Having this in the back of our heads, we then wanted to see how this correlates to brain structure. And we developed a protocol that would allow us to measure or to segment hippocampus to get an assessment of hippocampal integrity. And using this, Claudia Buss, a PhD student at the time, together with Michael Meany and Sonia Lupien in Montreal, contributed a paper in biological psychiatry in 2007, where she could basically show that the hippocampal volume has an effect that is associated with maternal care specific to females, here in conjunction with another risk factor for vulnerability for mental disease, birth weight. But this was interesting because this seemed to be specific to females. And we do know from the psychological disease literature that females are more prone, relatively 2-to-1, across the different psychological diseases to develop mental disease as compared to males. And then Veronika Engert again put this together to really say that, okay, can we make a picture out of this? And using a mediation analysis, she could show that with the parental care as a proxy of early life adversity, there was an effect on the cortisol response, as previously shown by Völlmin and herself in a cortisol response study. But hippocampal volume seems to mediate that, so that the perhaps parental care forms the structure in the central nervous system that regulates the HPA axis. And once the critical development periods are over, this then basically becomes crystallized and stays for the rest of the life. And that this is not restricted to the limbic system and the hippocampal volume we could show in a collaboration with Christine Heim, now professor in the Charité in Berlin, where we could show that another marker of early life adversity, childhood sexual abuse, also has programming effects on the somatosensory field, specific of the type of abuse, so that the formatting or the formation of the central nervous system in many aspects seems to be dependent on what happens to you during these early life areas. Okay, now finally, let's take a look at the effects in the body. So in the brain, take home, critical development periods, huge impact, which then also reverberates to the regulation of the HPA axis. But acute exposure to cortisol probably not doing that much damage in the brain. In the body, it's a little bit different. So there you do have very robust visible effects that you get from exposure to too much glucocorticoids. And this, I think, can be best summarized under the allostatic load model that was contributed by Bruce McEwen, where the basic argument is that you can maintain stability and functionality by adapting your body to these ongoing challenges that might be stress-related and therefore associated with chronic activation of the HPA axis. Since cortisol, as we heard, increases energy availability, then basically you have excess energy that often, given modern day civilized stresses, don't actually require extra energy. So you are no longer running away from a wild bear or a wild tiger coping with stress. You are trying to remember what the answers to the exam were in front of you, which really is a difference in terms of energy expenditure. So the excess energy that cortisol gives you now gets deposited, and typically deposited in fat stores. And perhaps the best example here is morbus Cushing, where you basically get these characteristic changes as a consequence of excess glucocorticoid exposure. And then glucocorticoids, cortisol in morbus Cushing is really a consequence of maximum HPA axis activation through a tumor. So here you basically see what cortisol can do if it's uncontrolled, ravaging through your body. And you have this characteristic buffalo hump, moon face, where you have fat deposition also in the face, a trunk obesity, where you have specifically weight gain in the abdomen and the trunk with thin legs and arms, thin skin, muscle loss in the periphery, redness of cheeks, immunosuppression, because cortisol is a very good immunosuppressant, and all that coming together. So allostatic load is the cumulative physical and mental wear and tear due to chronic exposures to stressors. So the physiological load that comes together with these increase in fat deposition is accompanying hypertension, the immune dysfunction, dysregulation of lipids, so the metabolic syndrome with the risk of adipositas and diabetes. And then the psychological load is a much higher likelihood for depression and mood disorders, correlating with the changes that also occur in the body, and probably also having to do with the fact that you feel constantly challenged by whatever, or threatened by whatever is happening to your body. And we did do a number of contributions also in the conjunction with the allostatic load model. Most of that contributed by Robert Paul Juster, who I was supervising as a PhD student together with Sonia Lupien at the Douglas Hospital in Montreal. He is now a professor at the University of Montreal, and he could show that you have, with high allostatic load index, this dysregulation of cortisol on the one hand, and a much higher incidence of burnout and depressive symptoms as a function or correlate of the height of your allostatic load index. He could also show that this seems to be gender specific, and that sex and gender roles do play a difference, or do have an effect here on the allostatic load. So as with the stress response being higher in men as compared to women, because of the estrogen, as outlined earlier, he could show that the overall allostatic load is higher in men, but this was further significant depending on specific gender roles, which was not present in women. And then finally, that this has 3 and 6 year progressive effects is something that he could also contribute by showing that the allostatic load index, that what was measured now, would still correlate with depressive symptoms 6 years later, even though not as concisely as the acute effect, but still significant, with 20% of variation explained by the allostatic load index 6 years prior. And that this cortisol has this metabolic effects, which in the long run then lead to this dysregulation of your metabolism and the accumulation of fat stores, is something that we have carried over now also to the work here. So basically, Maria Meyer, a postdoc in the lab, is doing a lot of work with the glucose manipulation. And interestingly, glucose drink just prior to a standardized stressor seems to amplify the cortisol stress response. So paradoxically, it seems to increase even more the effect that cortisol has. So adding bad to worse, if you like, perhaps best explained by the idea that you do need cortisol to, you do need glucose for cortisol to have its effects in the brain. And the idea here is a theory that would argue that cortisol is the insulin for the brain, because the brain is not insulin dependent, so it needs to depend on other agents for glucose to be transported, and cortisol might be just that. And that brings me to the end of this little potpourri of stress effects. If we summarize, what I think is important to take away is that psychosocial stress is a subjective experience. It's a perception. So you decide whether you are threatened or challenged to some extent by what's happening to you. The experience of stress causes the activation of body energy systems. And I think that is also very clear to help cope with the perceived increase in demand. So you get more energy to help you do that. Acutely, stress improves the encoding. So whatever happened that stressed you, you will better remember. But it impairs the retrieval, so what you've learned before, and now try to retrieve, bad luck. In the brain, acute stress is associated with a reduced activity of limbic system structures, importantly including the hippocampus, which might explain these memory effects. Chronic stress is not damaging to the brain, but the associated structural changes that have been robustly shown are more likely related to critical development periods that led to the formation in size of specific brain structures. In the body, however, there is wear and tear associated with chronic stress, mostly related to the metabolic effects. So you have these excess energy provision that leads to hypertension, adipositas, the metabolic syndrome, risk of diabetes, and then correlated with that much higher incidence of depression and mood disorders. And with that, I'm happy to take questions, but wouldn't want to stop before thanking all the funders over the years, and importantly also the cluster for their ongoing support for the work that we do, not primarily with stress now, but with physiological synchrony, which I think is just as exciting. And I'm looking forward to perhaps providing a talk on that topic sometime in the future. Thank you.
Very gladly, Jens, and thank you so much for this very fascinating overview and into your own research. Happy to take questions or pass around the microphone.
Thank you very much for the talk. It's great to see all the progress. I would like to understand a little bit more about the interpretation of the cortisol response, because it's needed, or it developed because it has some advantages. But then there is also the kind of progression to disease. And like with heart rate, they often see how long is a heart rate sustained after change, with blood pressure the same way. So is there research looking a little bit more into the, not the first half hour, but the first 3-4 hours, that you can actually stop it? And then you mentioned that a crucial difference is whether you can waste the energy or not. Mm-hmm. So is there a variation that people looked into, whether you sit here and do mental arithmetic or whether you have a chance to actually behave? So just curiosity.
Right. So maybe we start with the second question. I think the types of activities that are considered challenging to the system, where you actually use the energy. Let's say you run a half marathon. They are considered to be mostly beneficial to your physical integrity, even though you do get very strong activation of all the energy systems. And in the long run, this is supposed to make you stronger, healthier, more muscles, better, even though you have this. And if you are an athlete or a professional runner who does this very regularly, even though you get very regularly these high amounts of stress energy system activation. So that would be an argument for me to say that if you do use the energy, actually, it's probably a good thing. And then you can look at it as exercise. Now, you wouldn't want to look at psychosocial stress as an exercise because you get all this energy availability, but you're not using it. And I'm not sure that there's much research into that, but what could be one way to deal with that is to say, well, do a half marathon after you've been psychosocially stressed, so you can do away with that energy. And then maybe you can turn what normally is a threat to your long-term physical health into something that now is as beneficial as being a professional athlete. Of course, typically when you are psychosocially stressed, it also means you have a lot of work to do and there's little time in your schedule. And putting in a half marathon is not always easier said than done, but right, just thinking aloud. And the first question was, sorry, remind me. Oh, yeah. So you have, right, you have genomic effects that are being looked at where you basically get long-term effects from the glucocorticoid exposure, which are also investigated to say, what is it that is now the long-term effect? And you can look at this from the perspective of cortisol is the slowest, but the longest line of defense you have. So these genomic effects are also likely to then give you a sustained amount of change in response to this perceived change in demand. And this might have to do with the fact that you anticipate immune system activation necessary in a consequence of this threat that you perceived. You anticipate a fat deposition to become increased in response to this threat so that you prepare for the next time if you were now put in this alarming situation. There have been, I think, well, the one research I'm aware of was on long-term potentiation effects from glucocorticoid exposure that was contributed by Royal, I think, from the Netherlands. That would suggest that this is happening for a reason. So basically to give you a long-term adaptation to these increase or to this change in threat. Yeah.
So probably the social stress has been under evolutionary pressure, right? So why would you have increased energy then? What would be the evolution reason why we have cortisol increase in social stress?
With social stress? Yeah. So why is the social stress so potent in activating the HPA axis? That's a beautiful question, and there has a little bit of work on this as well, where basically the physical stresses give you a very robust activation of the autonomic nervous system. So I stand behind you and clap my hands unexpectedly for you. You will get a sympathetic activation from that, right? So it's very quick to respond because there's a physical threat. So your body might get harmed. Very robust activation of the autonomic nervous system. Cortisol will not react to that. You will need something that is socially relevant for the cortisol system to kick in. And the social relevance might also indicate that it's a longer term demand. Because when something is socially threatening, when you are at the danger of losing your social status, that is not something that is determined within the next 5 seconds, right? That is probably something that you now need a long amount of increased energy availability to make sure that you can preserve your status, that you can fight for the right place in the social hierarchy and not be rejected from your social peers. So it's considered to be a longer-term threat or a longer-term challenge when there's a social stimulus present. Does that make sense?
I mean, it's still unclear why you actually do need the energy, right? Maybe it looks like it's better actually maybe to have a good hippocampus to deal with the social situations better.
Right. Yeah. No. I do see the point, even though this is just something that I observed where we looked into what activates the HPA axis and saw this association. To me, the most sense is you will well remember what caused the activation of the HPA axis. So any event leading to that, you will probably muse about and ruminate about some time to come, so you can prevent it from happening again. So whatever caused you to make a fool out of yourself, if that is what constitutes a social threat, you are likely to think about for a long time to come.
Maybe a very basic question, but you showed like that the cortisol response is very, very robust. Is it possible to switch that? Because not everybody gets up at 9 because of people that work the nights. Do they have a shift in that, or do they completely work against the biology?
No, you do shift, right? And it's the same if you fly across the Atlantic. It takes a couple of days, and then basically you adjust to the new circadian rhythm. And with that, the cortisol awakening response shifts. So you will basically have the highest amount of cortisol when you now wake up 6 hours prior or 6 hours later, and you get the cortisol awakening response. It just takes a couple of days. And a lot of the hormonal changes that are associated with overcoming the jet lag are probably the reason why you have this experience of jet lag, because you need your hormones to readjust to the new rhythm. What you shouldn't be doing, however, is do that all the time. So I think that's one of the takeaways from shift work research, that it's okay to shift your rhythm once in a while, but don't do it every week, because then you basically screw up your hormonal system every time, and it can't catch up with these enduring changes that you impose on it.
I was wondering about the high and low stress responders. I mean, that's a physiological difference, right? I was wondering about the behavioral level. Do they actually cope also with stress differently? And is there something similar across the biological sexes? And might that be an influence on how presence of a partner has a different effect, because maybe male and female cope differently with stress?
Right. So let's talk about the sex and gender effects first. I think this result that you do get higher stress responses in women when they are supported by their male partner might have to do with the fact that males typically focus more on the competitive aspect of the situation, by and large, and females more on the social aspect of the situation. So when females support their male partner prior to the TSST, they basically focus on, just do your best, but no matter what happens, no matter how you perform, it's fine. I'm here with you. This doesn't happen anything to do with us, and then we are okay, and everything is going to be great. Whereas when males do the same thing, they might more act by saying that, you can do that. Math is not so hard. Just try to do the best you can. And that, of course, puts them in the completely wrong direction, and they get more stressed in anticipation of this task than they normally would have. So this is why this might have flipped. And of course, it depends on the specific gender identity that you have. So this is very overgeneralized or simplified, but I think this explains in part the result that we have seen here. Now, the overall magnitude response, does it have to do with the coping behavior? I think for early life adversity, I can tell you that the literature was for a long time divided on whether you get an amplification of the cortisol stress response or a blunting of the cortisol stress response after presence of early life adversity. So about 40% of the research showed that actually you respond more strongly, and then 60% of the research showed that you get a blunting. And the studies that we contributed in part also reflect this disparity, that depending on the circumstance, you get the one or the other. But for the ones that are more severely traumatized from an early adverse event that might even include sexual abuse, I think there you can basically make the observation that to them, the social stressor that we impose on them is actually not doing it. So it's no longer a threat to them because they've basically have seen much worse or have had other things to cope with.
Can I continue? So you mentioned that there was like about 50% people who are answering or responding to and the idea of 50% who do not respond
In the scanner.
Yeah.
Yeah.
You know, like, or what would be your thoughts about what are the contributors to that, and what would be the case, for example, in 500 years from now, after evolution appears? Like, do we go more into trying to cope better or less well with social stress?
Mm-hmm. Yeah, I think we evolved so fast that our stress response systems haven't had any chance to catch up with the situations that we are exposed to now that are threatening to our social self-preservation. And when you compare just a couple thousand years ago, we were basically living in caves in a very secluded small community, and now we are basically exposed to the world. One wrong social media statement and you know what can happen, right? So this is something where I think I'm not sure if evolution is as fast as 500 years. It might take even longer, but eventually it would probably adapt to that. Yeah.
So what do you think? There are 50-50 like answering it? It's just it's often like this
right. So
this robust.
The 50-50, I think on the one hand it has to do with whether subjects believed the cover story we told them, or whether they were basically saying that, well, I know I can do math a little bit and this is not normal. Also interesting is the Annie Duchenne, which I didn't present any findings from. A PhD student in the lab specifically looked at gender differences in the scanner stress. And she noticed that because women typically respond more to the social aspects of the task, they were less likely to respond to the stress task than men were. And she then changed the cover story so that she didn't say that, oh, this is a math task, even we want to see how good you can do math and please try the best you can. She presented it as, oh, this is for my PhD and I need this paradigm to work so I can complete. And then instead of saying you failed after 5 minutes, she came in and said, oh, it doesn't work again, I'm doomed. So it made the student or the test person bond with the researcher. And that made the women respond more in the scanner because they responded more to the social aspect of the situation, right? And she was able to manipulate it like that more effectively.